Somewhere between the prokaryotic and eukaryotic world, having characteristics of both, and the fact that many of these are the extremophil organisms, for example, the halophils or the thermophils, but also an important class of organisms are the various methanogens. And we'll come back and talk about methanogenesis when we talk about bacterial metabolism. In the domain bacteria, there are several phyla, and I don't expect you to memorize the names of all of these phyla. There are a couple you should know. And those are ones that are of quite a bit of interest to us and will be of interest when we talk about metabolism. And those are two phyla that constitute the phototrophic bacteria. So these are the photosynthesis bacteria. And the two phylum are the last two down here, the phylum Cyanobacteria and then the phylum Chloribia. So let's break down those a little bit, talk about those a little bit. Let's start with the phylum Cyanobacteria. What does cyan refer to? Yeah, it's a bluish green color. These used to be called the blue-green algae. What's wrong with calling them blue-green algae? Typically, what kind of cells make up algae? Eukaryotic. Okay, and these are prokaryotes. So they're now more commonly called the blue-green bacteria, but cyanobacteria is really technically the name of these particular organisms. You saw some of these in botany when you did the kind of survey of the plant kingdom. And at the beginning of the semester, I think you probably looked at a lot of the microscopic algae and things, and probably looked at Anabaena, and what was the other one you might have looked at? Maybe nostoc. Volvox is not a cyanobacteria. That's eukaryotic. Okay, that's a colonial eukaryote. These typically are long filaments and chains. You might have just looked at Anabaena. I don't know if that sounds familiar or not. So here is a slide that shows you some of the morphological forms of various blue-green bacteria. And sometimes some species come in these kind of packets. The more interesting ones occur in long-chain filaments. And what's interesting is these are not considered to be truly multicellular, but in Anabaena, I believe it is, there is a specialized cell within this filament called the heterocyst. Does that sound familiar at all? Yeah. Look at that. It was really pretty. It looked like a little inflated football. And this particular heterocyst is where the process of nitrogen fixation takes place. Do y'all know what nitrogen fixation involves? Nitrogen fixation involves taking atmospheric nitrogen and reducing it to nitrate. We're going to talk about nitrogen fixation a lot when we talk about microbial metabolism, because this is really an important metabolic process in agriculture. There are two types of nitrogen fixation. There is symbiotic nitrogen fixation and non-symbiotic nitrogen fixation. The blue-green bacteria are the non-symbiotic form. And we'll come back later when we talk about nitrogen fixation and talk about the significance of this heterocyst and why we need this specialized structure for this particular process to take place. Anybody know where symbiotic nitrogen fixation takes place? What does symbiosis mean? Literally means living together. There are two organisms that coexist with one another. And in that particular process, plant roots have nodules on them, and the nodules contain the bacteria. The bacteria fix nitrogen for the plants, And what do the bacteria get out of it? Food. The plants photosynthesize to transport sugars to the roots. So it's a win-win situation. What are the types of plants that carry out symbiotic nitrogen fixation? Have crops have nitrogen in the soil. Peas, clover, alfalfa, the legumes. Yep. Okay. So they're involved in the symbiotic nitrogen fixation. So this is a really important ecological phenomena, and these blue-green bacteria contribute to this nitrogen fixation process, but they do not have to be in association with another organism to do this out, so they can carry out the non-symbiotic. Another area where the blue-green bacteria can be important is oftentimes they are very early indicators of water pollution. I don't know if you've ever heard before, but sometimes in the summer, some of the reservoirs up in Indianapolis, they'll refer to them as having an algae bloom, and people can then get sick from drinking the water. Most of the algae bloom are actually the cyanobacteria, and they produce toxins as a result of their metabolism, and they produce flu-like symptoms. So sometimes when people say they have the summer flu, it's really toxin poisoning from these blue-green algae. And if too many inorganic nutrients like nitrates get into the water supply, that then fuels the growth of these blue-green bacteria, and they form these blooms where you can see it's scum on the surface of water when it gets really bad. That can also happen in farm ponds, where you've got fertilizer runoff into the ponds, and you can get all kinds of photosynthetic organisms growing, including these blue-green bacteria. But ecologically, where these are most important is these were the first oxygenic photosynthetic bacteria. So when we come back and talk about these in the context of metabolism, anything you learned and remember about photosynthesis from cell biology and the way higher plants photosynthesize, that's how these bacteria photosynthesize as well. Okay? They have two photosystems. Remember photosystem 1 and photosystem 2? And in photosynthesis, their source of hydrogen that they use to reduce NADP, sounding familiar at all? Is water. So they split water and evolve oxygen. Until the cyanobacteria evolved, the atmosphere of Earth was a reducing atmosphere. There was not free oxygen in the atmosphere. Okay? So the evolution of oxygenic photosynthesis had to precede aerobic respiration. You need oxygen in the atmosphere in order to support that type of respiration. I've already mentioned this, but the other significant thing that oxygenic photosynthesis does for us is that the oxygen that diffuses up into the atmosphere and now constitutes about 20% of our total atmosphere, under the influence of ultraviolet radiation, can interact to form ozone. And that protective ozone layer now then blocks the harmful forms of ultraviolet radiation that previously had made life impossible on the surface of the land. Everything had to be aquatic up to that point. So there's some very significant roles that the evolution of these blue-green bacteria or blue-green algae have played in the formation of the history of the planet. They literally modified our planet. Shut this off for just a second. I forget the name of this bay. This is in Australia, but it's a very popular site for microbiologists. If you go out low tide, there are all these kind of columns of blue-greenish material that look like rocks. These are called stromatolites. And I think there's a cross-section of one. Yeah, here's a cross-section of the stromatolite right here. These are huge biofilms made primarily by blue-green bacteria. And so if you can kind of think back, you know, a couple billion years ago when these organisms first evolved, the oceans of the world were probably colonized by these stromatolites, and the oxygen that they produced by oxygenic photosynthesis, bubble by bubble, diffused up into the atmosphere and really made our atmosphere an oxidizing atmosphere as opposed to a reducing atmosphere. So pretty important organisms. So if you go out and do some water testing, start looking at water samples microscopically, and there seem to be a large predominance of these blue-green bacteria in your water sample, that oftentimes is one of the very first indicators of the fact that that water source is polluted. Second phyla that we're interested in, so this is the phylum Cyanobacteria, is the phylum Chloribi. And we're going to use as our illustrative organism for the phylum Chloribi the green sulfur bacteria. Now, these are photosynthetic, okay? They have a different pigment than we find in cyanobacteria or in higher plants. This pigment is called bacteria chlorophyll. And we'll come back and talk more about what bacteria chlorophyll is and what role it plays in photosynthesis. But that's just kind of an association right now. This green sulfur bacteria have a different kind of photosynthetic pigment than bacteria chlorophyll. A really important thing about them is that they carry out a different type of photosynthesis called anoxygenic photosynthesis. So they don't produce oxygen as a byproduct. In this particular case, instead of oxygen, they produce sulfur. Why? Because they're not splitting water as a source of hydrogen. They're splitting hydrogen sulfide. And sulfur is produced as a byproduct. In some species, the sulfur is actually secreted outside the cell. In other species, that sulfur accumulates in granules. And we mentioned sulfur granules as one of the things that can be present in the cytoplasm of some bacteria. So these green sulfur bacteria have sulfur granules. So we're going to go back and dig into more detail the difference between these oxygenic photosynthetic strategies and the anoxygenic photosynthetic strategies. We're just not quite ready to go there right now. Okay, so there's a few things that you would find in Volume 1. The Archaebacteria, the Cyanobacteria, and the anoxygenic green sulfur bacteria. Volume 2. Volume 2 contains only a single domain. These are the protobacteria. And there's only a single phylum within Volume 2, and that is the phylum Protobacteria. And you'll notice that it is divided into 5 classes: alpha, beta, gamma, delta, and epsilon. We're going to mention a couple of those. We'll talk a little bit about class gamma and give you one example of the class alpha bacteria that has some interest and something that we'll talk about later on. Let's take a look at class gamma for a second. Okay? One of the most important families within that class, as far as you're going to be concerned, is the family Enterobacteriaceae. Also commonly referred to as the enteric bacteria. What does enteric refer to? Gut. Okay, these are oftentimes referred to as the gut-associated bacteria. We used to think that gut-associated bacteria were, for the most part, benign and had really very little effect on you. Then the emphasis switched to the fact that, well, there are some gut-associated bacteria that are potential pathogens. And that's why if those gut-associated bacteria that are pathogens are shed into water in the form of fecal material, and then somebody consumes contaminated water, they can contract disease from another individual. The article that I sent you that I didn't ask you to read at the beginning, but I'd like you to look at now, talks about some of the benefits that we're now finding from gut bacteria. There are several genera within this family that you're going to have occasion to encounter throughout the course of the semester. And some of your unknowns will be in these particular genera. The genus Escherichia, which is the E in E. coli. Salmonella. The genus Enterobacter. Or in the 7th edition, you may remember what's called? Aerobacter, okay? The genus Citrobacter. What's it called in the 7th? It was combined into Escherichia. Okay? It got its own genus in the 8th edition and beyond. Proteus. Shigella. Okay? There are several times when we'll be talking about these particular organisms. Serratia. All of these are gram-negative short rods. And sometimes they're so tiny and so short that they are confused by being cocci. So a lot of those are, anybody remember the term for a short rod that's kind of in between cocci and bacillus? Coccobacillus. So you have to be really careful when you have an unknown. And this is one of the reasons why I will verify for you, for your unknown, your gram reaction and your cell morphology. Because it really seems to me kind of tragic almost if you get your morphology wrong, because that's usually one of the first couplets in any key is Coccobacillus. Another important member of class Gamma that falls into this family Enterobacteriaceae is the genus Yersinia, and particularly the species Pestis. Here is a blood smear. Got some white blood cells here in the blood smear. Why can you tell these are white blood cells? Why do you know this is a white blood cell and not a red cell? Why do you know this is a white blood cell and not a red cell? What's this? What's this? What's this?
It's called granulocytes.
They are called granulocytes. But what are those cell structures that are stained red? They're nuclei. Okay, so why does that make it a white blood cell, not a red blood cell? Red blood cells are enucleated. Okay, they don't have a nucleus. So here are these little Yersinia pestis. This particular swelling is called a bubo, and it is characteristic of infection by Yersinia pestis. This individual is suffering from a disease that was notorious, particularly in Europe in the 13th century. I mentioned the fact that this particular disease affected about half the people or more and killed about a third of them. Kendall, which one? I see you saying it? Bubonic plague. Okay. There are other forms of plague. There's a pneumonic plague as well, but this is the Black Death. Okay. And we still will occasionally find cases of Yersinia pestis plague, but fortunately it's something that has been brought under control pretty effectively, particularly with the development of antibiotics. Another member of class gamma that's pretty interesting is the purple sulfur bacteria. Notice they have a different photosynthetic pigment than the green sulfur bacteria that we encountered in volume 1. These are also responsible for anoxygenic photosynthesis. Well, what does it say evolutionarily, the fact that green sulfur bacteria and purple sulfur bacteria are in different volumes of Berthius manual? Does that mean they're very closely related evolutionarily? No. Okay. So they split off and diverged quite early on and kind of took independent pathways. But they both evolved the same type of photosynthetic mechanism. And that's a very interesting thing about evolution, particularly when you've got 3.5 billion years of trial and error for organisms to evolve different strategies. These prokaryotes have been around for about 3.5 billion years. And that strategy of producing energy by photosynthesis was basically tried in different lineages of organisms. Okay, so that's why we can have these photosynthetic bacteria in two very different groupings, because they're not genetically related. They diverged very early on. In past editions of Berthius manual, where we didn't really depend upon information about genetic similarity, these would have been grouped together because they had the same functional process. The other class that we want to talk about just briefly with one illustrative organism within the proteobacteria is the class alpha. And these particular organisms called Rickettsia. Really interesting guys. They're very, very small. They are gram-negative. Most of them are pathogens, and they are a particular type of pathogen. They are an obligate intracellular pathogen. Does that terminology ring a bell from cell biology? Obligate intracellular pathogen. What's an example of an obligate intracellular pathogen we talked about in cell biology? No. What does obligate mean? Strict or absolute? Intracellular, obviously, have to live inside of a living cell. And pathogen means they cause disease. What do we talk about in cell biology that had to live inside another living cell in order to cause disease? Viruses. Okay. For a long time, people thought that these were viruses because of their tiny size and the fact that they couldn't live outside of existing other cells. The problem with these particular organisms, Rickettsia, is that they can't use glucose as an energy source. Did I tell you, they're going to fix it. They're coming in after class and trying to work on this. Actually, the energy source that these need to survive are various compounds that are part of the Krebs cycle. So alpha-ketoglutarate, succinyl-CoA, things like that. And you only find those in living cells. So these are sometimes referred to as being energy parasites. They can't really make ATP directly out of glucose. Why were they finally considered to be bacteria and not virus? Because they have both DNA and RNA. Virus have only one, not the other. And they have a peptidoglycan cell wall. A couple of diseases that they are notorious for causing are the disease typhus, and in this one that's called up here, Rocky Mountain spotted fever. These diseases can be lethal. One of the things that characterizes them is the development of a pretty significant rash. The reason that these cause a rash is that the pathogen has an affinity for endothelial cells. And one of the places you find a lot of endothelial cells are the lining of capillaries. Capillaries are a single endothelial cell layer thick. And when these cells infect the endothelial cells, they cause them to burst, and then the blood leaks out from those capillaries, and that is what gives that rash-like appearance. Rocky Mountain spotted fever is not confined to the Rocky Mountains. This particular disease, like most rickettsial diseases, are transmitted by ticks or other arthropods. So ticks, fleas, and mites are the primary vector or transfer agent for these rickettsial diseases. I did grow up in the Rocky Mountains. I grew up in Montana, and Rocky Mountain spotted fever was not uncommon when I was growing up. And you had to be particularly careful about checking yourself for ticks, because that's how the disease was transmitted. When they finally figured out that these were bacteria rather than virus, what new mode of treatment was then opened up for treating Rocky Mountain spotted fever? Antibiotics, okay. Previously, when they thought they were virus, antibiotics weren't even tried. The rickettsia life cycle is fairly complex, and one of the things that it does involve is a vector of some type, a transfer agent, picking up the bacterium from an infected host like a dog, and then the vector, the tick in this particular case, transmits it to the human. And Rocky Mountain spotted fever can have a variety of effects. I mentioned the rash, but it can cause fairly extensive vascular disease, and it can also then, if it can get into the cerebrospinal fluid, cause central nervous system damage and coma. So it's not something to be taken lightly. But members of this particular group, the rickettsia, are all transmitted by these fleas, ticks, mites, etc. Typhus is transmitted by, I think, both fleas and mites. There's another rickettsial disease that I'll just briefly mention called Q fever. And the context that we'll hear about for Q fever later on is that it is not uncommon to find these bacteria in cattle, and the bacteria can then be secreted into the milk of the cow. Pasteurization was a technique that was developed to try to prevent infectious agents from being transmitted in dairy products. And one of the things that pasteurization is designed to do is to kill this rickettsia that causes Q fever, because that's one of the more serious pathogens that are carried in milk. We'll talk about strategies of pasteurization, but it's kind of tricky, because in pasteurization what you need to do is you need to heat the milk to a point where it doesn't curdle and destroy the flavor, but you've got to heat it under the right conditions to destroy certain bacteria. And so this is one of the target bacteria we're looking at in developing pasteurization techniques. So many of you will find unknowns that fall into this particular volume if you have any of the Enterobacteriaceae. If you have members of this particular family, if you have one of those genera, Proteus or E. coli or Escherichia or something, they're really boring structurally. And when you start doing the staining test, you'll go, man, I don't get any positive tests for my bacteria. I know it's gram-negative, but it doesn't have endospores, it doesn't have this, doesn't have that. And sometimes people feel like they're not learning anything about their bacteria because it's not giving any positive results. A negative result for structural staining means just as much as a positive result. It's just as significant to know if it doesn't have something than if it does have it. These things really depend upon biochemical tests to start narrowing them down to species. And so the first part of the semester, you're not going to get a whole lot of useful information for your keying. But then you start doing your fermentation tests and all these other tests, and that's when you start really getting the information that allows you to differentiate between this whole huge, big group of organisms, the enterics, and narrow it down first to genus and then later to species. Okay, actually this is probably a pretty good place to stop because we're going to hit a new volume. So those of you who haven't had a chance to do your absorbance readings, try to get those done. Pull your plates out and either look at them and show them to me or throw them in the refrigerator. And then remember, we will have lecture tomorrow because you're having lab on Monday.